Hello everyone, and welcome to Biopedia. We are, in a sense, bringing back the Rewind mini-series today, because we will be discussing the fairly fundamental concept of cell division. Mitosis and meiosis might be two different concepts when it comes to eukaryotic cell division, but they'll be sharing an episode today. Both of these terms refer to forms of cell division. Because of this, we should clarify some of the genetics terms that will get tossed around here. These terms are chromosome, chromatid, and chromatin. Chromatin is a structure composed of DNA and protein. These condense into chromosomes, which are distinct units of chromatin. Once chromosomes duplicate to form the stereotypical X shape, the two versions within a chromosome are individually referred to as chromatids. With that out of the way, let's get to the main topics of today's episode. First, let's discuss mitosis. The term was coined by Walter Flemming, an anatomist from Germany. He first saw how chromosomes behave during this process in 1882. Mitosis is the process in which cells with a nucleus divide and end up with two cells identical to the one at the start of the process. Mitosis is just a fraction of the full cell cycle. 90% of the cell's time is spent in something called interphase. Interphase itself has three parts. The first of these is the G1 phase, or from my memory, growth 1 phase. The second is the S phase, standing for the synthesizing of DNA which takes place during this phase. The third of these three sections of interphase is G2. These three were initially thought to be pauses in the cell cycle, while in actual fact cells are growing, their chromosomes are being copied, and a lot of metabolism is happening during interphase. The remaining 10% of the cell cycle is when mitosis happens. There are 5 sections of mitosis: prophase, prometaphase, metaphase, anaphase, and telophase. During prophase, chromatin condenses into chromosomes, and the nucleoli also vanish. Mitotic spindles, made up of centrosome organelles and the microtubules that these generate, begin forming, while centrosomes move to opposite sides of the cell. We won't go through the centrosome today, but the microtubules are basically part of the cytoskeleton. The microtubules will be important in later stages, so keep a mental eye on them. In prometaphase, the nucleus breaks up and microtubules begin extending and moving into the nuclear region. Something called a kinetochore, made of protein, forms in the center of each chromosome. This is essentially where the microtubules, extending into the nuclear region, will attach to the chromosomes. I should say they've already duplicated at this point, as the S phase has already happened, and so they have the stereotypical X shape. In metaphase, chromosomes line up in the middle of the cell. You can visualize this as a line of X-shaped chromosomes in the middle of the cell, with the microtubules from either side of the cell extending towards them. The next phase is anaphase, where the chromatids in the chromosome are pulled apart. This is because the microtubule has now attached to the kinetochore and has begun retracting, pulling the two copies apart. The now separated chromosome copies each move to one end of the cell as the microtubules they are attached to become shorter. Next, in telophase, the nuclear envelope reforms. Chromosomes become less dense once again, and the two cells split. This relates to the next phase of cytokinesis. Something called a cleavage furrow forms in this phase. This is essentially the cell membrane pinching off so that one cell becomes two. Imagine it as a cellular figure of 8 shape before the two distinct cells form. I should clarify cytokinesis takes place in animal cells. That's mitosis done, so now let's transfer over to meiosis. This is a different form of eukaryotic cell division where the end result is not 2 daughter cells but 4. These cells also don't have all the genetic material of their parent cell, but only half. In other words, they are haploid rather than diploid. The difference between meiosis and mitosis is made obvious right at the start of the process. There is not one round of cell division, but 2. We differentiate between the first and second stages by adding a Roman numeral to the end of each stage. So, in prophase 1, genetic material is swapped between chromosomes. Instead of one line of chromosomes, as there is in mitosis metaphase, metaphase 1 of meiosis involves 2 lines of chromosomes. So basically, instead of one line of X-shaped chromosomes, there's a double line of them. These individual lines move into opposite cells, and two daughter cells then form. After the first round of cell division, these two daughter cells then undergo the process of cell division again. In metaphase 2, we get a single line of X-shaped chromosomes lining up in the center of the cell, but now half as many of them as we had in mitosis. These X-shaped chromosomes then split apart, and the genetic material then moves into opposite sides of the cell and into separate daughter cells. The end result of these two rounds of cell division is 4 haploid cells, with 2 cells having split to become 4 in the second round of division. Because of crossing over, which is the phenomenon of genetic material swapping between chromosomes which happened in prophase 1, these 4 daughter cells also have different combinations of the original genetic material that the parent cell had. This is because the goal of meiosis is to produce gametes such as sperm and egg cells, which will combine to form a diploid zygote upon fertilization. If you want a connection back to previous episodes, we discussed crossing over a bit back during episode 81. However, that brings us to the end of today's episode. Thank you all very much for listening. If you have any questions, comments, or topic suggestions for future episodes, feel free to get in touch at the show's email address. Until next time, have a great week, everyone.